Welcome to microbiology class. In this class, we will see how to identify bacteria in the laboratory. How will you identify bacteria in the laboratory? So for first identification, we require a pure culture. A previous video was there, how to prepare a pure culture. So first you require a pure culture for identification of bacteria. So once you have a pure culture, this pure culture can be identified by various methods. The first thing is colony morphology. Colony morphology. How is it appearing? Macroscopically, how it is appearing? What is the appearance? What is a colony morphology? Whether it is a dry, whether it is moist, whether it is mucoid, whether it is small or large, transparent, translucent, and what is the color of the colony. So that will give an idea about bacteria, but you cannot confirm the identification based on colony morphology. So first thing, you note down the colony morphology of the bacteria. Next thing, you do a Gram's staining. This is a common thing for microbiology, bacteriological identification. First you do a Gram's staining. So if you do Gram staining, you can identify whether it is Gram positive or Gram negative, Cocci or Bacilli. Shape also you can identify. Okay. So based on that you will get some idea. So after that, if you suspect a special motile or non-motile thing, you can do a motility test. Okay, you can do a motility test for demonstration of motility, hanging draw preparation or wet mount or mannitol motility medium or anything that can be used. Okay, so first colony morphology, then microscopic morphology is used. Next thing is biochemical test. Biochemical test. So there are various biochemical tests are available. So using this biochemical test, you can identify the bacteria. Test like MR, methyl red test, Voges-Prosker reaction, indole production test, H2S production test, urease production test, citrate utilization test, sugar fermentation test. So like that, there are various biochemical tests are available. So using this isolated bacterial colony, using this pure culture, you would perform this biochemical test. Unlike other chemical tests, the results of this biochemical test will be delayed. It may take time because most of them are enzymatic reaction. The bacteria you have to grow, and after when they are growing, they will produce enzyme, and that enzyme is present or not, that can be detected by this biochemical test. Okay, so that is biochemical test. There are various methods, various biochemical tests are available. After doing this biochemical reactions, you will get the result. So now we have this macroscopic colony morphology thing is there, microscopic Gram staining and motility details are available. Then we have this biochemical test. So based on these things, you can come to a conclusion that which genus is there and which species is there. And after that, you can confirm it also. There are some other, for that also there are some biochemical tests are available. So these are the common procedures performed for identification of bacteria. Apart from this, there are some serological methods also available for identification of the bacteria. You can detect the antigen present in the bacteria by antibodies. You should have in the laboratory, there are some anti-serum is available. So take that colony of the bacteria and put it in a slide and put your anti-serum, and you can see that reaction, antigen antibody reaction. You can see if it is present. If the antigen antibody reactions are taking place, you can confirm that this belongs to this bacteria. So that way also you can identify the bacteria. Nowadays there are some genetic studies are available. That is, we can identify the DNA gene of the bacteria by PCR methods. We can identify the gene. We can identify nucleotide sequencing can be done, and we can identify using nucleic acid probes, DNA probes. We can identify the particular DNA of the bacteria, whether it is present in this bacteria or not. Based on that also you can identify. Earlier days there was another method used for identification of bacteria was animal inoculation. they will inject this bacteria into this pure culture. They will be injecting into the bacteria into some laboratory animals, and they will see whether any particular disease is produced or not. So that was earlier days performed, but now animal inoculations methods are not used for identification. There are some special occasions are there, special things are there, in that they will be using animal inoculations. But otherwise, the identification of bacteria in the laboratory is mainly done by biochemical test. There are Catalase test, Oxidase test, and numerous biochemical tests are available. So using this test, we can identify the bacteria. Following this, you can see a video where they will be showing how to perform oxidase test and how to perform catalase test. Okay. Catalase is an enzyme produced by some bacteria. For example, Staphylococcus aureus. Staphylococcus aureus produce catalase. Streptococcus, they do not produce catalase. So both are Staphylococcus and Streptococci are gram-positive cocci. So to differentiate these bacteria, if you do catalase test, Staphylococcus are positive, Streptococci are negative. Okay, so that is one example of a biochemical test. Another thing is another example for biochemical test is oxidase test. Here also there is one enzyme. This enzyme, whether they are producing it or not, that can be detected by this oxidase test. For example, Vibrio cholerae, Pseudomonas, they are oxidase positive. Oxidase positive. Escherichia coli and other Enterobacteriaceae, that family bacteria are oxidase negative. So that way you can differentiate Vibrio cholerae, Pseudomonas from Enterobacteriaceae members. Okay. So these two things are as an example. They are demonstrating. This video will be demonstrating about these two biochemical tests. Please watch that and understand. This is only an example. Similarly, there are other biochemical tests also available to detect the enzymes and other chemical properties of the biochemical properties of the bacteria. Based on that, you can identify the bacteria. Okay, thank you.
Today, we're going to be learning how to do a catalase test. The catalase test is one of several basic microbiological tests used to differentiate bacterial species. How does it do that? Bacteria that contain the catalase enzyme can hydrolyze hydrogen peroxide into water and oxygen, and this will result in visible gas bubbles. It is especially effective in differentiating Staphylococcus, which is catalase positive, and Streptococcus, which is catalase negative. In this training program, you'll learn the proper techniques in performing two methods of catalase tests, the slide method and the tube method. For these procedures, you will need the following: personal protective equipment, hydrogen peroxide. A warning about hydrogen peroxide. A 30% solution of hydrogen peroxide is caustic. If skin contact occurs, wash immediately with 70% ethyl alcohol, not water. For these tests, a 3% solution will be used, but a higher concentration of hydrogen peroxide can be used for other bacteria. You'll also need sterile wood applicator sticks or plastic loops. Avoid using wire loops, as they can cause false positive reactions. You'll need pipettes and biohazard waste containers, and young colonies of bacteria on agar media. These colonies should preferably be 18 to 24 hours old. Use blood agar or chocolate agar media. If you're performing the slide method, you'll need 2 sterile glass slides and 2 sterile Petri plates to contain aerosols produced by the catalase reaction. If you're performing the tube method, you'll need 2 12 by 75 millimeter test tubes and a test tube tray. These tests are usually performed on the laboratory bench. Because these tests produce aerosols that can carry infective bacteria, you should perform this test inside a Petri dish, a tube, or inside a biosafety cabinet. Now let's review the slide method. Place each slide, one for the positive reaction and one for the negative reaction, onto the bench top. Label them and place them in separate, uncovered Petri dishes. Using the wooden stick or plastic loop, touch the center of an 18 to 24-hour-old isolated colony, place on the glass slide, and discard the applicator or loop into a Sharps container. If you're using colonies grown on a blood agar plate, be careful not to pick up any red blood cells with the colony. Red blood cells contain catalase and can provide a false positive result. Repeat the process with your negative control. Next, using a dropper, add 1 to 2 drops of hydrogen peroxide solution to the growth on the positive control slide and immediately place the cover on the Petri dish. Against your benchtop or a dark background, look for the formation of bubbles immediately upon the addition of the hydrogen peroxide. An immediate formation of bubbles indicates a positive reaction. Discard your pipette into a biohazardous waste container. To verify the accuracy of your reagents, next, test the negative control slide. Using a dropper, add 1 drop of hydrogen peroxide solution to the growth on the negative control slide and close the Petri dish. No formation of bubbles indicates a negative reaction. To perform the tube method, first label the two 12 by 75 mm test tubes with positive and negative and place them in a test tube tray. The tube will contain the aerosols. Then add 4 to 5 drops of hydrogen peroxide solution to each of the 12 by 75 mm test tubes. Discard your pipette into a biohazardous waste container. Use the applicator stick or plastic loop to pick up a small amount of 18 to 24-hour-old isolated colony from the positive control. Place and leave the applicator stick or plastic loop in the tube. Place the tube against the benchtop or a dark background and look for the immediate formation of bubbles. Repeat the process for the negative control. No formation of bubbles indicates a negative reaction. So as you can see, the Catalase test is a rapid, straightforward test that when done properly can differentiate between Staphylococcus, which is Catalase positive, and Streptococcus, which is Catalase negative.
In this training program, you'll learn the proper technique to perform the oxidase test. This test is one of several tests used to identify gram-negative bacteria. The oxidase test is based on the production of the enzyme endophenol oxidase or cytochrome oxidase. Endophenol oxidase oxidizes phenylendiamine oxidase reagent to form a dark purple compound, endophenol blue. This is usually only present in aerobic organisms. You have a positive reaction if the paper develops a deep blue color within 10 to 30 seconds. This indicates that the endophenol oxidase has oxidized the phenylendiamine oxidase reagent to form endophenol blue. For this procedure, you'll need the following materials and equipment. Personal protective equipment, timer or clock, sharps container, biological waste container and bag, disposable Petri dish, filter paper. Do not use the filter paper once it begins to darken. Sterile wooden sticks. You can also use platinum loops, plastic loops, or platinum non-nichrome wires. For the reagent, we'll be using an oxidase solution based on the formulation by Kovacs. There are multiple methods used in the clinical laboratory for performing the oxidase test. In this video, we will only show the filter paper method. Please note, you should always refer to the reagent manufacturer's instructions. This procedure is made up of 3 steps: preparing the filter paper, testing the colony, and interpreting the results. First, place a piece of filter paper in a Petri dish. Next, moisten the filter paper with 1 or 2 drops of oxidase reagent. To test the colony, pick an isolated colony with a stick, wire, or loop and smear onto the reagent-dampened filter paper. place the stick or plastic loop into the sharps container. If a wire or platinum loop is used, then sterilize it according to your protocol. Set the timer and observe for no more than 60 seconds. You have a positive reaction if the paper develops a deep blue color instantly or within 10 to 60 seconds. You have a negative reaction if the paper does not show any color change within 60 seconds. The oxidase test is one of several tests used to identify gram-negative bacteria. There are two possible results with this test: a positive reaction which shows a blue-purple color within 10 to 60 seconds, and a negative which shows no color within 60 seconds. Now we should dispose of all the lab waste into the biological waste container.